Hello and welcome to another Piper Pod. Today we're going to start a brand new series. It will probably be about a 20-part series, and we're going to look at the complexities and individuality of wound care. Now, like any good series, we need to build a good foundation of understanding on the topic before we move on. So today's lesson is going to focus on the physiology of wound care itself. Before we press into that, I think it's apt of me to do a little bit of a review on the anatomy and physiology of the skin itself. Now, the poor skin is often forgotten in a lot of anatomy physiology talks. It's a very, very important organ, and one that is often forgotten that it is an organ. It's in fact our largest organ and can take up about 15% of our body weight and hold up to a third of all of our blood as well. Now, the skin is very important for a physical effect, but also a psychological effect. It's often referred to as the body's mirror, as it reflects the person's physiological health, their age, their ethnic group, their backgrounds, any cultural differences. So it has a larger importance than a lot of the other organs. It's incredibly complex, as most things are in the human body, and it's filled with a lot of supporting apertures, if you will. Now, in a single centimetre square of skin, you can have 15 sebaceous glands, almost a metre of blood vessels, 100 sweat glands, about 3,000 sensory cells and ends of nerve fibres, about 4 metres of nerves, 15 pressure apparatuses to record tactile stimulus, about 200 nerve endings to record pain, 2 sensory apparatuses for cold, about 12 for heat, about 300,000 epidermal cells, and 10 hairs. So all of that is happening just in a centimetre square. Now, the skin can easily be divided into 3 distinct layers. Now, I know you're well aware of this, but the epidermis layer is the outer layer. It contains a couple of protective cellular structures which basically don't have a nuclei, and the nucleus only starts at your stratum granulosum, which is the middle layer of the epidermis. Now, this is what pretty much feeds the top 2 layers, and as it progresses up to your stratum seposium, the next level up, it loses the nuclei itself. So enzymes will actually destroy it, and this is where the cell structure starts to give stability and elasticity to the actual substance itself. Now, an important point to note about the epidermis, one of the most important points, is known as your basement membrane. So looking at the picture, if you see the area between the epidermis and the dermis, which is also known as the dermal-epidermal junction layer, you'll see that it's not a straight line. It goes up and down and pretty much follows a contour based on needs around certain apertures themselves. Now, the basement membrane is a good separation of non-vascular, non-inverted membrane that separates the 2 layers. It's semi-permeable membrane which regulates the transfer of your proteins and other minerals such as oxygen and nutrients across this junction. So this actual junction is one that allows us to maintain our moisture, allows us to not essentially leak out as we do with burns, and it's a very important layer that also feeds the rest of the epidermal cellular layers themselves. Now, the next layer down is the dermis. Now, this is not as thick as people think, and obviously it's the middle layer. Now, this can best be described as the factory of the skin. Okay, and it's pretty much broken up into 2 different layers. You have the papillary layer, okay, and then you also have the reticular layer. Now, the papillary layer lies just underneath the basement membrane zone, and it's 1/5 the size of the dermis. Now, its main role is the facilitation of the nutrients and oxygen to that basement membrane. So it has what's known as a papillary loop, which will actually supply the oxygen up, allow things to travel down to the dermal layer, and pretty much be that junctional gap between the dermis and the epidermis that keeps that basement membrane alive. When we move into the reticular layer, this is where you find your collagen fibers. Okay, and the collagen fibers are found in layers of the dermis. The thicker the layers, the tougher and more flexible the skin. Okay, it allows it to stretch and accommodate with increased weight. Now we're going to talk a lot more about collagen fibers later on, so we'll move on to other functions found within this reticular layer. This is a layer where all the activity starts. So obviously it contains that collagen, but it also has two plexus of nerves which control your exocrine sweat glands. It contains the foundation for leukocrines, prostaglandins to allow chemotaxis to occur. It's responsible for your natural heparin and histamine and control of vasodilatory response in certain physiological states, which we will look at. This layer of the dermis is also the support framework for the epidermis and the attachment for the hypodermis below. And if that wasn't complicated enough, it also has several sensory receptors found within this dermis. Now the hypodermis is the largest layer of the skin. Okay, it's also known as the subcutaneous layer, and this is basically just a main support framework of the skin, and it allows for attachment of protective layer for the underlying organs and structure. It's comprised mostly of adipose tissue and connective tissue with blood vessels. The other function of the hypodermis is temperature regulation and storage of lipids themselves. Before we move on to the extra physiology of the skin, I just want to spend a little bit of time talking about a structure that gets overlooked but is very, very important in overall skin health, and that's the extracellular matrix. So this matrix is very complex structure that surrounds the cells in all tissues, and it has its biomechanical compositions that vary between the tissues. Now in healthy skin, this extracellular matrix is a gel-like matrix and constitutes the largest compound of the dermis itself. So we talked about the dermis and what was found in it. It's mostly this extracellular matrix that makes up the dermis and lies between those cellular structures themselves. Now within this extracellular matrix we have compositions of polysaccharides, water and collagen proteins, and as mentioned they're responsible for the skin's elasticity, tensile strength and compressibility as well. Now these skin characteristics are due purely to this extracellular matrix, and in particular the molecules that are found within it. Now the function of the extracellular matrix is to act as mechanical support for cells and tissues. It integrates those cells and tissues as well, and it also influences the shape and any cellular movement that occurs. It influences the development and differentiation between the cellular structures, and it acts as your reservoir for the extracellular signaling molecules. And it also regulates and directs key wound healing processes. So the reason we're focusing on it, the reason we're moving into a little bit more details on this extracellular matrix than you may have had before, is because every physiological effect of wound healing to come is pretty much centered around the dermal layers and this extracellular matrix support. So moving into the physiology, wound healing can pretty much be broken up into two stages. We have haemostasis and then wound repair and regeneration. So haemostasis is the first step, and it's designed to rapidly control hemorrhage and start the healing process. So the way it works, it's all got to do with destruction to the endothelial lining in between the layers, which support that extracellular matrix where the collagen is found. So once you have endothelial damage, initially what occurs is you have natural vasoconstrictors coming into play. So you have your catecholamines and your serotonin that cause a overall vasoconstrictive response in order to minimize blood flow to that area. On top of that, when the collagen fibers are exposed to the outer layers, or the lumen where the actual platelets are found, it actually attracts the platelets, and the platelets get stuck to the collagen fibers. Now, when the platelets stick to these collagen fibers, the platelets will actually release further vasoconstrictive mediators in the form of prostaglandin. At this point, it's very important to note that serotonin and prostaglandins are class terms of body mediators, and you'll find differing effects that they occur. In this particular case, the prostaglandins release will cause vascular spasm, which will further reduce the blood flow. On top of all this happening, the platelets are also responsible for releasing phospholipids and adenosine diphosphate, which increases the amount of platelets and actually attracts platelets to come, causing a plug. Now, the plug or lot itself is then encased with fibrogen and fibronectin, or even a substance known as vitronectin, as well as a few other factors like von Williamsburg. The main part is this clot forms what's known as a provisional extracellular scaffold that allows cellular migration of the inflammatory cells and fibroblasts to be brought towards it. Now, platelets also release cytokines, hormones, and chemokines, which also attract inflammatory cells. This influences the wound healing phase. The entire biochemical response of this haemostasis involves the formation and degradation of a blood clot. Now, it does this through your intrinsic clotting pathways as well as your extrinsic pathways, mixed with clot reactions and a process that you'll all be aware of, which is fibrogen lysis. The complex process involves many different clotting factors, 13 in total, that release as forms of damaged tissue. The cycle sees the development of a clot, the reaction and compaction of that clot, and then finally the breakdown of that clot by fibrogen lysis, and it moves into the rest of the healing phases. The first phase is known as the inflammatory phase. Now, this happens pretty much at the same time as hemostasis, and even other times without hemostasis when you have a wound that doesn't involve bleeding. Now, it lasts from impact, if you will, tissue injury, up to around 4 days. And it's usually divided into early and late stages. Now, just breaking it down, it has a few different pathways which generally start up and set a platform in order to move on to healing. So the first one we've sort of already mentioned in the case of hemostasis, and that's the release of chemical mediators. Now, these are different sort of mediators than your hemostasis, but they fit within the same class. So there's things like complement proteins, different types of prostaglandin, and histamines response. Now, these cause vasodilation as opposed to vasoconstriction and vascular spasm, which occurs in your hemostasis. It also increases the capillary permeability and attracts neutrophils, monocytes, and different lymphocytes to the area by creating a chemotaxis. So basically creating a chemical signal in order for those cells to migrate out of the bloodstream into the extracellular matrix. In the presence of your vasodilation, you get hyperemia, and that's where we get our heat and we get our redness, or two of our cardinal signs of inflammation. Now, the increased permeability of the capillaries causes extra exudate to form. So the capillaries leak all their fluid, which creates an exudate. Now, in lessons to come, we will look at all the different types of exudate and what those pathological characteristics actually mean. In a simple sense, this fluid leaking exudate leaks this rich protein into the tissue space, which has a large osmotic draw. And that large osmotic draw increases the amount of fluid in that area. That's where we get the swelling, and most of the time that's where the pain and immovability of joints come from as well. Now, in the presence of these cardinal signs, we obviously have an increase of blood, which increases the metabolic rate of the cells because of the temperature, and increases the amount of oxygen and nutrients that are available in the immediate area. The increased amount of protein in that space will bring a lot more nutrients and fluids to the area, as well as substances used to support the haemostasis at whatever stage they're in, whether it's the generation of the clot by providing fibrogen or the presence of plasminogen to aid in the fibrinolysis. Now, whilst all this is going on, we also have secondary effects that are occurring during the inflammatory stage, which is to do with white blood cells numbers. So when the tissue gets injured, as well as releasing chemical mediators associated with vasodilation, leukocytosis inducing factor. Now, leukocytosis means that this has increased the amount of red blood cells that come to the area, and it migrates to that particular sites. So we have our chemotaxis. Most of the chemotaxic signals come from the release of this leukocytosis inducing factor. Now, the migration of these cells obviously occurs in order to clean up. Okay, so the leukocytes will pass through the capillary walls, and they'll go to the area, and they'll go through phagocytosis of any pathogens, dead tissue, cellular debris, whatever's there. In the short term, you have neutrophils. Neutrophils are there quickly, and then in time, your macrophages will actually turn up. Now, when the macrophages turn up, that's when the whole physiology changes. Okay, so we have an increase in the amount of pus, so exudate turns quite pussy, and it also has the probability of generating a fever based on infection as well in the presence of these macrophages. Now, sometimes in the macrophages' presence, we get a little bit of pus formation, cleans up the area, and then we move on to our healing side of the house. Now, the healing phase is a very, very long phase, and it's broken up into two large components. So we have proliferation, also known as reconstruction phase, and then we have remodeling, which is also known as maturation phase. Now, proliferation phase we'll discuss first, and this generally occurs halfway through your inflammatory phase, so about day 2, all the way up to around about day 24. Now, the main purpose of this phase is to regenerate new tissue. Okay, so it's done with a series of 3 different types of cellular formation. So you have your fibroblasts, your angiogenesis, and your epithelial cells. Fibroblast proliferation is stimulated by the release of growth factors and other mediators in the inflammatory stage. So fibroblast is mostly concerned with producing fiber-based structures. So myofibroblasts, your collagen, and basically anything that gets deposited within that main matrix is your fibroblast side of the house. Now, with this stage within proliferation, we get the ability for the wound to contract in on itself, and it also increases the strength around it. So instead of having a clear cut, these cables, fibric cables, will actually come together and they'll pull, increasing tactile strength, and they'll pull the wound close, and increases the strength of it, closes the gap, and decreases the probability of it reopening. Now, this is also the formation of scars. Now, this happens on top of the skin, but it also happens with wounds as well. So any state of inflammation, the damaged tissue will be replaced with these cable-like structures. So it doesn't actually get replaced by healthy cells and all the rest of it. It's more just being replaced with these cables. And that's the tissue that actually gets formed, as opposed to cells just remultiplying themselves in this stage. So most of the gap that fills in a wound is actually done by creating this matrix of pretty much collagen-based fibers. Now, the ones that do produce new cellular formation is your angiogenesis. So this is the formation of your endothelial cells and new blood vessel formation. So blood vessels will regenerate and will regrow within those areas. Now it is actually the macrophages we spoke about binding to that extracellular matrix that release a growth factor which stimulates this collagen synthesis and fibrogen proliferation. It also stimulates the need for angiogenesis. Now the last thing that occurs during this proliferation or reconstructive phase is re-epithelialization. So this is where the epithelial cells will actually migrate over the granulated wounds. So granulated wounds meaning the wounds that's created, that scar tissue after the contraction of the wound bed has occurred. Now the epithelial cells will migrate from the surrounding wound edges, okay, or even from hair follicles or sweat or sebaceous glands within the wound itself. Now on appearance, they appear as this really thin, translucent film across the wound, and they are very, very fragile and can easily be removed by pretty much anything that isn't just gentle cleaning. A lot of the time, using the wrong dressings will actually destroy this epithelialization and prolong the proliferation phase itself. The final stage of the whole healing process is that remodeling or maturation phase. Now this involves, believe it or not, the actual reduction in capillaries and the amount of those collagen and other fibrous-based scar tissues. Now, the original collagen that was produced during the proliferation phase is disorganized and it's not very strong, so it needs to be replaced. So during this remodeling phase, the collagen is slowly replaced and in a more organized manner. Now, this results in increased tensile strength and a considerable decrease in the vascularity and the size of the scar itself. Okay, so that was a pretty heavy overview of wound healing. So we'll just finish off with a bit of a summary just to help get a good foundation. Now a lot of these things obviously we're going to refer to when we look at specific wounds, specific wound dressings, stuff like that. So it will be good to have a reasonable understanding of the things that we spoke about. But just to break it down, haemostasis occurs in the presence of bleeding. Okay, so once we get a break in the vascular wall, we need to go through haemostasis, which is mostly controlled by the release of mediators, collagen moving into the blood space, into the blood vessel, and attracting platelets. From platelets forming a clot and then working hand in hand with inflammation. Now, inflammation differs from haemostasis because it causes vasodilation instead of vasoconstriction and vascular spasm. Now, during the inflammatory phase, we also have recruitment and activities of other inflammatory cells. We end up with those classic inflammatory signs, which is your erythema, your heat, your swelling, and your pain, all due to inflammatory processes. Partway through inflammation, we start moving into proliferation. Now, this is our tissue repair initially. So this is the provisional extracellular matrix is denatured. Okay. From there, angiogenesis occurs and granulating tissue or scar tissue is formed over the top. After which we get wound contraction and re-epithelialization occurs. When we transition from proliferation into remodeling, we start that remodeling of the wound and gradual increase of tensile strength because the collagen fibers that were disorganized and quite weak are individually replaced by stronger and longer collagen fibers, which increases its overall strength. Okay, so that completes the first lesson of the series. Lot to take in. Hopefully you got something out of this. It is encouraged that you go back and review this lesson, have a look at some textbooks, get a good understanding of the foundations before moving on to the other lessons. The next lesson, we'll start looking at the specifics of wound healing, and we'll look at different modes and aberrants of healing itself. But until next time, take care.